I'm Daniel Levine, and this is the Bio Report. Antibodies have been powerful tools for inhibiting a targeted protein. Abalone Bio is pursuing a new class of antibody therapies called activating antibodies that can regulate cellular processes and restore their balance. One aspect that makes these rare antibodies attractive is that they can target previously undruggable G-protein coupled receptors, allowing them to treat diseases that have been hard to address. We spoke to Richard Yu, co-founder and CEO of Abalone Bio, about activating antibodies, how the company generates massive data sets for its AI-driven platform technology, and how it looks beyond binding to explore the function of these antibodies. Richard, thanks for joining us.
Thanks for having me. Really exciting.
We're going to talk about target activating antibodies, Abalone Bio's AI platform technology, and its efforts to develop a new class of antibody therapies. Let's start with antibody therapies as they exist today. In general, how do these work to treat disease?
Yeah, so the antibodies have more or less clustered into a few buckets, right? So there are those that take a hint from their natural functions in our body, which are to, identify, cruise around, take out invading pathogens, right? So things that bind and block and or otherwise take and direct the immune system towards whatever they're binding, right? Then there are molecules that have been engineered to take advantage of some of the characteristics and then sort of extend the capabilities. So there are ways of using them to shuttle, or payload, deliver payloads to a site of action. So, antibody drug conjugates or ADCs, of course, very well known and quite popular these days for, delivering a toxic payload to a cell you want to destroy, or even bispecifics where antibodies have these multifunctional structures so they can bind a couple things at once, or can be engineered to do that. So you can have one head that'll bind a target cell, and the other head that'll bind something that you want to bring to the target cell, like a T cell, for example. There are many therapies along those lines. And then in the third bucket, there are antibodies that act like actual pharmacological agents, right? So in addition to sort of like blocking a receptor or perhaps blocking, binding and soaking up a signal that would essentially act like block a receptor. You have ones that can also have pharmacological activities. They can bind, activate, or otherwise modulate the signaling properties of the target that it's bind.
While these have been powerful therapeutic tools and great commercial successes, drug developers have been limited in the targets they could pursue with them. Can you explain why?
Yeah, well, drug development is just hard, first of all. It makes sense to sort of focus on the simplest, mechanisms of action first, right? You pick the low-hanging fruit. Something where antibodies that are really good at doing, just binding something specifically, you want to make your first crop of drugs that do that, bind. So when you think about those types of drugs, it's like, how is it binding? Where is it binding? How tightly? And then, thinking about how does the rest of the overall molecule behave as a drug? Is it something that's soluble enough, or can you get it at a high enough concentration? Is it stable enough? Can you make it so that a person can inject this at home with a small needle instead of having to get an IV infusion? All those characteristics you have to consider. I think in terms of what's been done in the past, we also are sort of led by the technologies, the discovery or development technologies, right? So for antibodies, we've started off taking advantage of animal immune systems and immunizing them with proteins or fragments of proteins of targets of interest that we would like to find antibody binders to, right? And then later on, we had other in vitro technologies, phage display or yeast display, where you have ways of producing some target of interest, some protein of interest, whether it's in a protein that's affixed to a bead or the surface of a glass plate, or on the surface of a phage, or on the surface of a yeast cell, and then displaying antibodies on the phage or the yeast to see if they bind to the protein that's been fixed onto a surface or a bead, right? So if you do that, you are really focusing on things that bind tightly, right? So there's this affinity focus. And I think that limits you to the types of targets that are, good for that type of modality, right? So targets that have a very distinguishable large blob that's sticking out, if you're talking about a receptor that's sitting on the surface of the cell, that can be easily recognized and bound to by an animal, right? So I think, sort of historically, we weren't led by what is the actual best target we want. It's more like, hey, what can we actually make with the technologies that we have available? And then sort of go from there and see what targets are most addressable by its technologies.
There's been a lot of interest in the potential to reach so-called undruggable targets and GPRC targets in particular. What makes these so compelling?
Yeah, well, GPCRs are on, they're a major class of cell surface receptors, right? They're like the eyes and ears or nose of the cell, right? So they're the things that listen to chemical signals and then mediate the cell's responses to them, right? So, smell receptors, taste receptors, light receptors, those are all GPCRs, or G protein coupled receptors. I should spell the acronym out as it's first mentioned. So and of course there are some very important GPCRs that respond to hormone signals or other chemical signals like GLP-1 that everyone has heard of at this point, right? These are the major class of drug targets. Oft-cited statistics are something along the lines of about a third of approved drugs target GPCRs, and before the explosion of Ozempic, Wegovy, and the like, they're already heading towards a couple $100 billion market, and now that's probably going to be essentially doubled by the obesity drug market. But what's curious is that even though they have been so successful, actually most of them have not been successfully drugged, right? So only about a quarter of them have drugs developed for them. So sort of breaks down into a few reasons. Specificity can be an issue, right? Since these are so prevalent and often cluster into different subtypes. You have like 5 somatostatin receptors, right, that are pretty close to each other, right? So often you can find a small molecule or a peptide or non-antibody drug that can do what you want it to do against that target. But what happens is you light up a whole panel of GPCRs, right? So you get these off-target effects, right? And then actually some of these targets are quite complex, and it is hard to get activity of interest with a small molecule. Some of them have, larger extracellular domains, and they need to wrap around a large protein-based signal. So chemokine receptors are like this, where it's a large protein-based signal, and then multiple parts of the extracellular parts of the GPCR need to interact in just the right way with the signal, the chemical signal, to have an effect. So a small molecule may not have the sort of surface area or the chemical complexity to induce, to bind all the places that it needs to bind to induce the right sort of conformational changes to activate the thing. So, what makes them compelling is just that, they're a fundamental part of biology, right? Conserved from yeast to humans. I think they make up like 4% of the genome, something like that. So, biology's devoted enormous resources to keeping them around for a good reason.
So you mentioned activation. A lot of people think of monoclonal antibodies as inhibiting, and you're focused on antibodies that activate and modulate cell surface receptors. Can you explain what it means to activate a receptor?
Yeah, I think, at a super sort of cocktail party conversation level, it's always easier to gum up the works, right? So in terms of inhibiting, it's easier to get in the way of something, right? You bind and you either sort of lock it, give it a big bear hug, so you block a receptor from being able to receive a chemical signal, or just otherwise get in the way of its natural activities. Activation is more of a problem, particularly for G protein coupled receptors, right? So GPCRs have historically been quite challenging to find antibody agonists for. These are highly dynamic proteins. There's 40,000 atoms and 500 amino acids roughly per protein, goes in and out of the cell membrane 7 times, so it's this really complex, dynamic protein. And for them to activate, what they need to do is, they're always jiggling, right? But when they bind their natural chemical signal, they start jiggling in a different way, right? They promote or tend to adopt another set of conformations, an active state conformation. And that allows it to interact with appropriate downstream proteins to sort of carry forth signals and have the cell respond appropriately. So it's a lot, if you were to look at just sort of a random sort of antibodies, it's just statistically way less frequent to find an antibody that binds a target in just the right way to nudge this jiggling, mass of atoms in the right way to activate it, as it would be to find something that sort of gets in the way of something, right? So that's the big problem. It's like a very sparse and a difficult to solve problem.
So do activating antibodies exist in nature, or is this an abalone creation?
Yeah, it's interesting. There actually are a number of GPCR antibody activators, so-called auto-activators, that are essentially disease causers, right, or they're disease-related, right? So they're, in the billions of humans that have existed through history, our immune systems are usually good about keeping and filtering out antibodies that bind to our own proteins. But occasionally some slip through, and then occasionally from that, the subset, some of them will bind to a target in a way that activates it, right? And some of those are GPCRs. So yeah, there are, I think, roughly 3 or 4 dozen described auto-activating antibodies. And it pointed the way to us to say, hey, it is possible for us to find or otherwise design antibodies that can bind to these targets in a way that can activate them. And just another statistic throw out there is, as far as we're aware of, there are only 6 GPCRs that have ever had antibody activators intentionally developed for them. So, we've done 2 of those too, right? So this is like, all of biotech history, right? So these are pretty rare molecules, right? But it is possible. And we're finding that, if you measure enough, and now it's sort of the modern tools that are available to really extract patterns from enormous amounts of data, you can actually start designing some really cool molecules that have the pharmacological properties that we want.
Is there any way to quantify how these activating antibodies may be able to expand the targets available to go after?
Yeah. I mean, I think, there's always known targets that have known liabilities, right? So it's like, we have some preclinical or even clinical data showing that if you activate this target, you can get some, helpful effects in patients. But unfortunately, we have these side effects. And if you can narrow down the side effects to, for example, off-target effects, hitting another receptor, that those targets become, are put back on the table, right? So in that sense, it's sort of like expanding the universe. So we're not really trying to necessarily just be a longer lasting drug, for example. Though that's a perfectly good strategy for some companies. Antibodies can be engineered to last quite a long time in the body, as opposed to other drugs. But we are not focused. We're really focused on, hey, can we access targets that have historically just been very difficult or impossible to drug? And does it make sense to use an antibody to do that in the context of the disease that we're trying to address?
In addition to being able to go after targets that were inaccessible to monoclonal antibodies, my understanding is these have other desirable qualities. What are those?
Yeah. I mean, antibodies in general, by virtue of their chemical complexity and their large size, are very specific, right? So specificity is one of the major check boxes for and benefits for antibody as a modality, antibodies as a modality. They are also genetically engineered or genetically encoded drugs, right? So the engineering can use genetic engineering techniques. So you can mix and match parts quite quickly. Modules. So you can search sort of drug space quite efficiently. They're also, as I mentioned earlier, have a good pharmacokinetic properties, so they can be engineered to last quite a long time in your body. So having drugs that can have a 2 to 4 week dosing schedule can be really good for patient compliance, and also just for chronic disease in general. You don't have to worry about pills every day or injecting yourself peptide every day or every week.
The company has developed an AI platform known as its functional antibody selection technology, or FAST, to identify activating antibodies. How does the platform work?
Yeah, so at a high level, we're really co-opting a couple key features of nature, right? So there's the ability to self-replicate, right? And then there's this algorithm of survival of the fittest, right? And we use those to measure ton of activity data on antibodies to find the rare ones that actually do what we want them to do, right? And then with that data, we can take advantage of the modern computational tools like machine learning to really dig into the patterns of sequences and their relative activities and learn how to develop better molecules, right? So the way I try to think about it is that it is a giant search problem, right? So you got like 100 million keys to try to search through to open up a lock, right? And only a few of them will do that. So how do we do that, right? So analogously, I think conventional technologies are really focused on, hey, can we build robots to pick up a key and put it into the lock faster and test it that way? Or hey, can we miniaturize these things? Can we make smaller versions of these locks and keys so that we can test more of them using the similar amount of reagent? So these are all what I'd call like conventional sort of high throughputizing technologies, right? And what we want to do is take advantage of those properties of biology, right? So just sort of extending this analogy further, what if we taught the locks how to copy themselves and duplicate themselves, and then have each of them both make and then test one of those 100 million keys on itself, right? And then only if that key opens up that lock, will that lock be able to reset itself and then further test and divide, right? Otherwise, the lock stays shut and goes dormant, right? And that's what we've engineered yeast cells to do, right? Saccharomyces cerevisiae, the same sort of workhorse of synthetic biology that is also brewing your beer and baking your bread, right? So what we've done is we've engineered these quasi-human cells to both make our GPCR target of interest. It has the DNA encoding it. It puts it onto the cell surface, cell membrane, just like it would be in our human cells. And then it gets, through some engineering, connected to existing yeast signaling pathways inside that allow it to transmit signal into the cell. Right. So then from there, we can take that signal and connect it to the expression of a growth promoting gene. Right. So the logic goes something like, you have a receptor that's active and that's functional in the cell, so that if you have more activity of the receptor, you get more signal cruising through the yeast signaling pathway that produces more growth promoting gene, and then you get more cell division, right? So in these cells, we then also make sure that each of them makes and tests a single antibody variant. We put antibodies on single copy DNA elements, we put them into these cells, and then the cell will produce the antibody in addition to the target of interest, sort of put it out on the outside of the cell in a way that the cell, the antibody can interact with the receptor. And then if the receptor is activated, that signaling pathway gets turned on and that cell divides and makes more copies of itself, right? And we can do this all in very, in massively parallel fashion. Because another cool thing about these yeast cells is that they're like these quasi-plant cells. In addition to being quasi-human cells, they're also quasi-plant cells, right? So they have a cell wall on the outside of them, which is this sort of mesh that prevents large proteins from going in and out of the cell. So it creates this little isolated atmosphere around every single yeast cell. What that does is it essentially makes every yeast cell this sort of very, very small test tube, isolated test tube, and we could grow them all next to each other in a big pot. And those antibodies are only testing themselves on an individual cell basis, right? So that's really, really powerful because then we can cram, a billion of these cells, each of which is minuscule, 50 femtoliters or so, orders of magnitude smaller than even the smallest digital BCR droplet or something like that.
And all of this is
grow them together.
All this is being done in a wet lab as opposed to doing this in silico?
Yeah. So this is all wet lab. We are primarily wet lab because it's the data on these antibody sequences and how they function against the receptor, which is the key. And that is the bridge too far, at least now, for sort of predictive AI capabilities. We've been very good about going from antibody, or let's just say protein sequence to structure, and we're getting better now about predicting how those structures can interact with each other. But that sort of final mile of figuring out those specific interactions to perturb the structure in a way that activates them or otherwise modulates their signaling capabilities in a way that's interesting is not there yet, right? Unless you have large amounts of data, which we are generating for different receptors and different receptor discovery campaigns, then you can start getting more generative and start mapping out the landscape of sequences, antibody sequences, and how they function against these UPCRs. And then you can take advantage of the ML tools to sort of extrapolate to new sequences that may have improved properties.
And are you using existing libraries, or are you doing something to generate potentially novel antibodies?
Yeah, we're doing both, right? So we're starting off with naive human or animal libraries, and then we encode them genetically into these cells. It's quite flexible. We can take any genetically encoded library of, frankly, any proteins. They don't even have to be antibodies. We can do other proteins or peptides if you would like. But we are focused on antibodies. So we start off with those, and then we use our data sets to identify hits, and then we can take those learnings, and then we can essentially do an analogy of traditional sort of maturation or directed evolution, or we can take hits and generate small diversities around interesting molecules and then further test those. We can supercharge that the benefit of our platform is that allows us to supercharge that process. And much like phage display or yeast display, they have these abilities to do affinity maturation, right? So those things are, again, focused on affinity, and they will bind some population of phage displaying antibodies to a target, for example, wash with some stringency, take those off and then rebind them and then wash with an even higher stringency, right, to get ever higher affinity antibodies. We can do a similar thing with our platform too. We can do what we call a functional maturation, right? So we can essentially twiddle the knobs on the cells that the sort of threshold of signaling that the cell needs to have before it starts turning on that growth promoting gene, right? Then we can sort of find ever more potent antibodies from that. And we've demonstrated that we can do that with the hits that we have.
Abalone's lead program is a CB2 agonist for the treatment of pain, inflammation, and liver fibrosis. This would be a potential non-opioid to treat neuropathic pain. What's the opportunity and the need there?
Yeah, we're very interested in that neuropathic pain space because there really is no good drug out there, right? If you talk to neurologists, we have many quotes that say, in general, things really don't beat the placebo when you look at the actual numbers. Diabetic neuropathy, which is probably the largest market, is huge. 7+ billion patients in the US alone, and nothing is really working, right? So the various GABA modulators or serotonin or norepinephrine drugs or opioids, for example, they all sort of do something to be squint right, but none of them really are effectively treating the symptoms, nor are they disease modifying, right? And that can be really debilitating for these patients because those who are suffering from the neuropathy, it's not just tingling in your fingers. It's someone described it as essentially a room temperature butter knife, feeling like you're grabbing something dipped in liquid nitrogen or equivalently to heated to 1,000 degrees, right? It's like it just literally causes pain, both pressure and temperature variation. So it's quite debilitating. There are the obvious huge problems with opioids. So looking for things that are non-addictive is a huge driver here. And it just turns out that the target that we're going after, the cannabinoid receptor CB2 for this disease area, has good, ample preclinical evidence to show that it's very good for neuropathic pain.
And what's known about your experimental candidate from studies you've done to date?
Yeah, we have shown activity on in mouse models of a related indication, a chemotherapy-induced peripheral neuropathy. So we have gone through a set of molecules. We've identified a lead, and we're optimizing that into a development candidate now. We've also expanded the studies into its use in liver fibrosis and MASH. So we're we've seen some anti-inflammatory activities in precision cuts human liver slices, and which are indicative of some sort of therapeutic effect in a MASH scenario. And then we're doing some animal studies right now. So CB2 sits at this intersection of a lot of interesting biological properties, not just analgesia, but also just inflammation in general, and also fibrosis, right? Direct activities on anti-fibrotic activities. So agonism of this target has a widespread potential benefit, and antibodies really are just like a great use case for this target. CB1 is the main off-target receptor. That is, of course, the primary psychoactive receptor for THC. I usually joke around, call it the Pink Floyd receptor, right? So you really want to avoid cross-reactivity to that. So not only just from the psychotropic effects, but also because activation of that actually has sort of like pro-inflammatory and pro-fibrotic activities in certain circumstances. So off-target activity is not just unpleasant side effects, but actually like directly undercutting your therapeutic mode of action. So specificity is very, very important for targeting CB2. And, we have made some of the small molecules or bought them as controls for both our in vitro and also in vivo studies. And even ones that are purportedly, thousandfold specific for one over the other, you do see evidence of cross-reactivity when you jam them into a mouse. There's a high peak effect. There's also, different distributions and whatnot. So within the organism. So yeah, I think what makes it so beautiful for antibodies is that, they're binding in a place that the normal chemical signal doesn't for CB2, right? So, the cannabinoids all sort of bind in a similar pocket between CB2 and CB1. By looking at, if you look at the crystal structures of them, they basically overlap perfectly. But if you look at the outside of the protein where the antibody bind, they are very different. It's 25-ish% sequence identity. So they're very, very divergent at the outside because these regions of the receptors weren't really involved with any sort of function, so they weren't under selective pressure to maintain any homology. So yeah, that makes it a perfect use case for antibodies. We're binding in an outside place, which is highly divergent, and we're able to, massage the structure of those jiggling atoms in the way that would allow it to activate and have some of these pain, pressure and cold sensitivity reduction effects.
And what's the development path forward?
Yeah, well, we're primarily just focused on generating a good product right now, working towards a development candidate. Most of the action that we've seen to date has been from partners, interest in that end. Though, of course, there's been quite a bit of activity in the pain space at large. The NaV1.8 non-opioid drugs are getting quite a bit of interest in action lately. So perhaps there's a resurgence of that market, sort of nicely parallel to the sort of resurgence of the MASH market as of late. Both pain and MASH have sort of been, known as graveyards for a while, and we'll see if there's a turnaround there.
What are you looking at beyond the CB2 therapy?
Yeah, we see a lot of really interesting opportunities in addressing underserved or unserved populations in really large markets like obesity, right? So we're really quite interested in a sort of next generation obesity drug now that would allow us to get the benefits of some of the incretin-like drugs, suppression of appetite and satiety, without the GI side effects, which are intolerable to ever larger fractions of the GLP-1 drug taking population, right? So in this case, there's so much clinical benefit to reducing food intake and reducing weight that the markets are so large that even a 10, 20, 30% of the overall global obesity market we're talking very, very huge numbers and immense potential for impact, right? So we have identified some targets where if you could hit specific subclasses of the targets, we can do that because we can take advantage of the specificity that our antibodies can afford, then we would be able to activate specific neural circuits that would allow us to separate sort of good parts from the bad parts of some of the existing weight loss drugs. So that's really exciting. In addition to that, there are a number of other targets that are on our short list in all sorts of therapeutic areas. I mean, GPCRs are active across all areas of biology, and everywhere from cancer to immune-related diseases or autoimmunity. So those are areas that we're also quite interested in as well.
Given the platform technology and your focus, it would seem there is a big opportunity to partner around potential therapies. How are you thinking about the role that would play in your business model, and how do you think about balancing partnerships with your own internal drug development?
Yeah, this is probably a very common experience for therapeutics discovery platform technologies or companies that have a sort of discovery technology at its core. We're focused primarily on products. I mean, products are our North Star. Not only is it just personal interest to actually develop, things, getting them closer to the patients, but also the platform is only as validated as your sort of latest stage product, right? So the overall balance will evolve over time. We started off with doing more discovery-based partnerships, like, hey, you're having trouble with this target. We have a technology that might be able to address that. Let us help you with that, and we can sort of share on the upside. And then now we're more focused on developing internal projects that we can develop to a certain point, and some of those we can either drive further down the development pipeline, potentially even to the market, or we can license them out or otherwise monetize them at later stages when they've created much more value. So we're at that point where we're sort of transitioning from one to the other. It's not binary, of course. It's more a change of the ratio of the two. So we're still interested in doing, high value, sort of discovery partnerships for the right targets where it makes sense.
The company's raised about $22 million to date. How far will that take you, and what's the plan for raising additional capital?
Yeah, so that's a mix of investor capital and grants and partnership revenue. We've used primarily to both sort of build out the platform and do proof of concept work and do sort of initial partnership activities. Our next round that we're doing is to drive the development of our pipeline. So we're really excited about these metabolic/obesity programs and driving CB2 further. And so it's all about pipeline at this point for the next round.
And what are you finding discussions with potential investors like these days, given the current environment?
Very much product focused. Think the sort of heyday of the 2021, 22, you have a cool technology and a dream and a vision, and we'll throw, large amounts of money at you, are not really here right now. That's okay. We've been just, heads down and just trying to find the molecules that we want to find for the targets that we think where we can have the best and largest impact. That being said, it does seem like things are wakening up a little bit. Let's call it a thaw or a détente or something in the marketplace. So yeah, we're having some productive conversations with investors right now that we're excited about what the near future will bring.
Richard Yu, co-founder and CEO of Abalone Bio. Richard, thanks so much for your time today.
Yeah, no problem. Thanks so much for the opportunity to chat. Appreciate it.
Thanks for listening. The Bio Report is a production of the Levine Media Group. To automatically download this podcast each week, subscribe to our RSS feed or through iTunes or other podcast manager. To join our mailing list, go to levinemediagroup.com. We'd love to hear from you. If you want to drop us a line or are interested in sponsoring this podcast, send an email to danny@levinemediagroup.com. Special thanks to Jonah Levine, who composed our theme music, and the Jonah Levine Collective, which performs it.